Welcome back, everyone, to another deep dive. This time, we're taking a closer look at Mycobacterium tuberculosis.
A fascinating bug, to be sure.
Absolutely. And for this deep dive, we'll be referencing Principles and Practice of Clinical Bacteriology, 2nd edition, by Gillespie and Hockey.
An excellent resource.
Really is. So for all you medical microbiologists out there, get ready to test your knowledge, because we're going deep into the weeds today. We'll be talking about what makes this organism unique, how it causes disease, and some of the historical perspectives that might surprise you. Sound good?
Sounds great.
Perfect. Let's jump right in. So what makes Mycobacterium tuberculosis so special? What sets it apart from other bacteria?
Well, for starters, it's not just one bacterium. It's actually a whole complex of closely related organisms. Genetically, they're over 99.9% similar.
Wow, that's incredibly similar.
It is. They're more alike than humans are to chimpanzees.
Amazing. You'd almost think they were clones.
Almost. But despite this remarkable similarity, they do have some fascinating variation.
Okay, I'm intrigued.
Well, one of the key things is their ability to adapt and evolve. Unlike a lot of bacteria that depend on point mutations, M. tuberculosis primarily adapts through what are called insertion sequences. These are little bits of DNA that can move around within the genome.
So like jumping genes.
Yeah, kind of. And these jumping genes can cause additions or deletions of whole genes.
So it's less about subtle tweaks and more like rearranging existing genetic material.
Right. Imagine shuffling a deck of cards. You get a different arrangement each time, but you're using the same cards.
Interesting analogy. And I imagine this has some important implications for how we think about the evolution of M. tuberculosis.
Definitely. It's changing our understanding of the bacterium's family tree.
Speaking of family trees, I remember learning that M. bovis, the one that causes bovine tuberculosis, was thought to be the ancestor. Is that still the case?
Actually, no. Recent research, specifically looking at those genetic deletions, suggests that M. africanum or even M. canneti might be the real ancestors. It's kind of like a detective story, piecing together the evolutionary history.
Fascinating. Shows how our understanding is constantly changing. But let's move on to how this bacterium actually causes disease. What can you tell us about the pathogenesis of tuberculosis?
Well, it all starts with the bacterium entering the lungs and encountering the first line of defense, macrophages.
Those are like the body's first responders.
Exactly. They're immune cells that engulf and destroy invaders.
So they try to gobble up the M. tuberculosis.
Right. But M. tuberculosis is a clever little bugger. It's developed ways to survive, even thrive, inside those macrophages.
No way. How does it do that?
Well, for one thing, it prevents the macrophage from becoming acidic, which is normally how they kill bacteria. And it also manages to stay within the macrophage's recycling pathway so it gets access to nutrients.
So it's basically hijacking the macrophage, turning it into its own little safe house.
Exactly.
Pretty sneaky, huh?
Yeah, that's impressive. But the body doesn't just give up, right? What happens next? Well, it sends in reinforcements. More immune cells, like monocytes, arrive on the scene. They transform into more macrophages. But even with all these extra troops, they still can't initially kill the mycobacteria.
So it's kind of a stalemate.
Yeah, you could say that. And the stalemate can go on for quite a while.
So how does the body finally get the upper hand?
T cells, the elite forces of the immune system. They show up and release interferon gamma.
Interferon gamma.
Yeah.
Rings a bell. What does it do?
It's a powerful signal that activates the macrophages, finally giving them the power to destroy the bacteria.
Ah, so the T cells are like the strategists directing the attack.
That's a good way to put it. And they're not just directing, they're also supplying a key weapon, nitric oxide.
Nitric oxide. That's a powerful molecule, right?
It is. Along with other radicals, it's essential for killing M. tuberculosis.
So it's this complex interplay of immune cells and molecules that finally controls the infection.
Precisely.
But I imagine the story doesn't always end there, right? What are the possible outcomes of this battle?
Well, there are two main paths. The infection can be controlled and become dormant. It essentially goes into hiding within the body. Or the mycobacteria win and spread further, leading to active tuberculosis. And that's when we start to see the clinical features of the disease.
Clinical features. Now we're talking. So how does this microscopic battle manifest itself in patients? What are some of the signs and symptoms that we see?
Let's start with the most common presentation, pulmonary tuberculosis. Picture a patient coming in with a persistent cough, maybe a low-grade fever, just feeling generally run down.
Those are pretty vague, aren't they?
They are, and that can make diagnosis tricky in the early stages. As the disease progresses, though, they might start losing weight. And in severe cases, they might even have trouble breathing.
Okay, so those are the classic symptoms. But I imagine they could easily be mistaken for other respiratory infections, right?
Absolutely. That's why we rely on diagnostics like chest x-rays.
Chest x-rays. Essential for getting a visual of what's happening in the lungs.
Exactly. Not just for confirming the diagnosis, but also for seeing the extent of the disease. Are there cavities, pleural effusions? We need the whole picture.
Makes sense. And I'm guessing newer imaging techniques like CT scans give even more detail.
They do, and sometimes we might even need to do a bronchoscopy.
Oh, wow. That's where they insert a thin tube into the lungs, right?
Yeah. It lets us look directly at the airways, and we can take samples for testing.
Okay, so that's pulmonary TB. But I know tuberculosis can be a real trickster. It can affect other parts of the body too, right?
Yeah.
What about those less common extrapulmonary presentations?
Right. It can spread to all sorts of places. One of the most serious, though thankfully rare, is tuberculosis meningitis.
Meningitis. That sounds scary.
It is. It's an infection of the lining of the brain and spinal cord.
What are the symptoms like?
Well, imagine someone coming in with a really bad headache, a stiff neck. Maybe they're drowsy and confused.
Sounds like a bad flu.
It can mimic other illnesses, which makes it tricky to diagnose. But if it's not caught early, it can progress really quickly, even to coma.
That's terrifying. How is tuberculosis meningitis diagnosed?
By examining the cerebrospinal fluid.
And that's done through a lumbar puncture, right?
Exactly. We look for things like white blood cells and protein, which are usually elevated in meningitis. And we can also do cultures and molecular tests to try to detect the bacteria directly.
So it's kind of like a puzzle, putting together the clinical picture with the lab findings to make the diagnosis.
It is. It's not always easy. And sometimes the number of bacteria in the CSF can be really low, making them hard to find, even with molecular techniques.
I can imagine that would be frustrating.
Definitely. But it just highlights the importance of a thorough clinical evaluation and a high index of suspicion.
Good point.
Yeah.
What about other extrapulmonary manifestations?
Well, lymph node tuberculosis is another common one. It can affect lymph nodes anywhere in the body, but we usually see it in the neck.
Swollen lymph nodes. I'm guessing that could be caused by a lot of things.
True. But lymph node TB is particularly prevalent in areas where tuberculosis is endemic.
Endemic meaning?
Where TB is regularly found in the population. And it often presents in children.
Okay, that's helpful to know. So how do you distinguish lymph node TB from other causes of swollen lymph nodes?
Sometimes we need to do a biopsy and then examine the tissue under a microscope. And of course, we'll do cultures and molecular tests to look for the bacteria. But TB can also affect bones and joints.
Really? Bones and joints? I never would have thought of that.
It can. In fact, we even see evidence of spinal tuberculosis in ancient Egyptian mummies.
Wow. So TB has been plaguing humans for millennia.
It really has. It's an ancient disease. And it's been a challenge to treat for centuries. In fact, did you know that before antibiotics, one of the standard treatments for TB was fresh air?
Wait, what? Just fresh air, like going outside?
Pretty much. There was a whole movement called the sanatorium movement.
Sanatoriums?
Yeah.
Those were like hospitals specifically for TB patients, right?
Exactly. And they emphasized rest, fresh air, and good nutrition as the main treatment.
It sounds almost too simple to be true. Did it actually work?
Well, it did help some people. The focus on rest and good food probably helped to boost their immune systems, which allowed some of them to fight off the infection.
That's amazing. It shows that sometimes the simplest things can be the most effective. Of course, once antibiotics came along, everything changed.
Oh, absolutely. The discovery of antibiotics was a game changer for TB treatment. The first truly effective drug, streptomycin, was discovered in 1944.
Streptomycin, the first weapon in our modern arsenal against TB. But I'm guessing the story of antibiotic resistance is also a part of this journey.
Unfortunately, yes. Resistance to streptomycin emerged fairly quickly, especially when it was used alone. This really highlighted the importance of using multiple drugs or combination therapy to prevent resistance from developing.
Combination therapy, a strategy that's still a cornerstone of TB treatment today. So what other drugs were discovered after streptomycin?
Well, PAS was another early drug, but the real heavy hitters, the ones that really made a difference, were isoniazid, discovered in 1952, and rifampicin in the 1960s.
Isoniazid and rifampicin. Those are definitely familiar names to any medical microbiologist. How did these drugs change the game?
They drastically improved treatment outcomes, and perhaps most importantly, they allowed us to shorten the duration of therapy significantly. We went from years of treatment in sanatoriums to just months of treatment at home.
Wow, that's a huge difference from isolation to outpatient care, all thanks to these powerful antibiotics. But I'm sure ensuring patients actually completed their treatment became a new challenge.
Absolutely. That's where DOETS, or Directly Observed Therapy Short Course, came into play.
DOTS, a brilliantly simple yet effective concept. But for those who aren't familiar, can you explain why it's so important?
Sure. Imagine you're a patient and you start feeling better after a few weeks of treatment. You might be tempted to think you're cured and stop taking the medication.
It's understandable, right? You feel better, so you think you're good to go.
Exactly. But the problem is those TB bacteria can be sneaky. If you stop treatment too soon, they can come back with a vengeance. And they might even develop resistance to the drugs, making them much harder to treat.
Well, that's scary. So DOTS is all about preventing those relapses and the emergence of drug resistance by ensuring patients take every single dose as prescribed.
Precisely. It involves having a healthcare worker, a pharmacist, or even a trusted family member watch the patient take their medication each time. It's like having a cheerleader who keeps you motivated and on track to beat TB for good.
DOTS, such a simple yet powerful intervention. But unfortunately, we know that drug resistance continues to be a major challenge in the fight against TB. And speaking of drug resistance, what are some of the unique challenges in managing drug-resistant TB, particularly MDR-TB?
MDR-TB, which is resistant to at least isoniazid and rifampicin, is a very serious form of TB. The mortality rate is much higher, and treatment is longer, more complex, and often involves more toxic drugs. It's a whole different ballgame.
It sounds incredibly difficult to manage. So how do you approach these cases?
It requires a highly individualized approach. We have to tailor the treatment regimen to each patient, considering their specific strain's drug susceptibility profile, their overall health, and the potential side effects of the drugs. There's no one-size-fits-all solution.
Individualized care. It makes sense, especially when dealing with such a complex and potentially life-threatening condition. So what are some of the guiding principles in managing MDR-TB?
Well, first and foremost, we need to know exactly which drugs the bacteria are resistant to.
Right. No point in using drugs that won't work. So how do you determine that?
Through in vitro susceptibility testing. It's crucial. We can't just guess or assume.
Data-driven decisions. I like it. What else is important?
Combination therapy is still key, but we use at least 3 drugs to which the bacteria are known to be susceptible. And in areas with high rates of resistance, we often start with a 4-drug regimen right off the bat.
Hit them hard and hit them with multiple weapons. I see the logic there.
Yeah.
And does DOTS still play a role in managing MDR-TB?
Absolutely. Adherence to these complex regimens is even more crucial for MDR-TB. We need to support patients every step of the way.
DOTS, such a simple yet powerful tool. Are there any other considerations when managing MDR-TB?
Yes. We have to be very cautious about adding new drugs to the regimen. We only do it in pairs, and only if we're certain the strain is susceptible to both drugs. Otherwise, we could inadvertently fuel further resistance.
It's a delicate balancing act, trying to stay one step ahead of those adaptable bacteria. But I'm curious to hear more about these specific mutations and how they lead to resistance to different drugs.
Sure. Let's take rifampicin as an example. Most cases of resistance to rifampicin are due to mutations in a specific gene called RPB.
RPOB. Okay, I'm following. What does that gene do?
It codes for a subunit of RNA polymerase, which is an enzyme essential for bacterial survival. These mutations alter the binding site of rifampicin, essentially preventing it from working properly.
So it's like the bacteria are changing the locks on their doors so that rifampicin key no longer fits.
Exactly. Very clever of them, isn't it? And another common example is isoniazid resistance.
Isoniazid, another first-line drug. What causes resistance to this one?
Well, one common mechanism involves mutations in the KG gene, which codes for the catalase enzyme. Isoniazid actually needs to be activated by this enzyme to work properly. But if there are mutations in KG, this activation process can be disrupted, rendering the drug ineffective.
So it's like the bacteria are cutting the power supply to the isoniazid weapon.
That's a great analogy. It's incredible how these seemingly small genetic changes can have such a huge impact on treatment. But remember, it's not just about the bacteria and their mutations, it's also about us, the medical microbiologists.
You got that right. We play a crucial role in identifying these resistant strains. But I'm curious, even with all the sophisticated technology we have today, there must still be challenges in diagnosing TB, right?
Oh, absolutely. TB can be a tricky bug to pin down, especially when it comes to extrapulmonary cases or when we're dealing with patients who have weakened immune systems.
So what are some of the challenges you face in the lab?
Well, for starters, those nucleic acid amplification tests, or NETs, which have been a game changer for rapid TB diagnosis, particularly for respiratory samples.
Nets. They're fantastic, aren't they? They can detect the bacteria's DNA directly in the sample.
They are, but when it comes to non-respiratory samples, like biopsies or cerebrospinal fluid, things get a little trickier. The bacterial load in those samples is often much lower, so the sensitivity of the NET can be reduced.
I see. So careful specimen selection and result interpretation are key.
Exactly. We have to take into account the type of specimen, the patient's clinical presentation, and other factors to ensure we're interpreting those results correctly.
And how about molecular susceptibility testing? Can we reliably predict resistance based on genetic mutations alone?
For most anti-TB drugs, it's still difficult to do that with complete confidence. Resistance mechanisms can be complex, involving multiple genes and pathways.
It's not always a simple one-to-one relationship between a mutation and resistance.
Right. But there is one exception. Rifampicin. Remember how I mentioned that over 90% of rifampicin resistance is due to mutations in that small region of the ARC-POP gene?
I do. It's like a beacon, a telltale sign.
Exactly. We can use techniques like direct sequencing and commercial kits that specifically target this region to quickly identify rifampicin resistance. It's been incredibly valuable, especially in managing suspected MDR-TB cases.
It's amazing how far molecular diagnostics have come. It's like we're able to peer into the inner workings of the bacteria and decipher their secrets.
Sure.
But even with all the advancements in technology, at the end of the day, it still comes down to people, right? The patients, the clinicians, and the lab staff all working together to tackle this complex disease.
Absolutely. It's a team effort. We in the lab have to communicate clearly with the clinicians, explaining the limitations of different tests and helping them interpret the results.
It's about connecting the dots and seeing the bigger picture, right?
Right. Exactly. And speaking of the bigger picture, let's shift our focus to the epidemiology of tuberculosis. What are some of the things that make this disease so challenging to control on a larger scale?
Okay, epidemiology it is. So we know TB is primarily spread through the air, like through coughing or sneezing.
Yeah.
But what factors actually influence how easily it spreads?
Well, one factor is the intensity of the cough. Someone with a really forceful cough, especially if they have laryngeal tuberculosis, is going to spread more of those infectious droplets.
Right, makes sense. Like a sneeze versus a gentle cough, the sneeze will spread those droplets much further.
Exactly. And certain medical procedures, like bronchoscopy or even autopsies, can generate aerosols containing TB bacteria.
Oh, wow. Hadn't thought about that.
Yeah, that's why proper infection control in healthcare settings is so important.
Absolutely. We need to protect healthcare workers.
Right.
But I'm guessing not everyone who breathes in these droplets gets infected. What determines who actually gets sick?
Well, it's multifactorial. It depends on things like how many bacteria they inhale, the virulence of the particular strain, and of course, the individual's immune system.
So like a perfect storm of factors.
You could say that.
And are some people more susceptible to developing active TB than others?
Definitely. People with weakened immune systems, like those with HIV, are much more likely to get infected and develop active TB.
HIV and TB, a terrible combination.
It is. Unfortunately, the HIV epidemic has actually fueled a resurgence of TB in some parts of the world, particularly sub-Saharan Africa.
That's heartbreaking to see these two epidemics colliding.
It really is. And then you add the long incubation period of TB to the mix, and it gets even more complicated.
Right. That's a time between exposure and when symptoms actually appear, right?
Exactly. And with TB, that can be weeks, months, or even years.
Wow, it's like a ticking time bomb.
In a way, it is. And it makes tracing outbreaks and identifying everyone who might have been exposed incredibly difficult.
I can only imagine. So how do we track the spread of TB and identify these potential outbreaks?
Well, contact tracing is important. That's basically identifying and following up with people who've been in close contact with someone who has TB. But in recent years, we've also seen the emergence of molecular epidemiology as a powerful tool for tracking the spread of TB.
Molecular epidemiology, using DNA fingerprinting to track strains of TB. Pretty cool.
It is. We can use these techniques to compare different isolates of TB, see how closely related they are, and even trace the origins of outbreaks.
So you're like detectives, but at a molecular level. Amazing. And have there been any specific strains that have really caught your eye?
There's one that's gotten a lot of attention. The Beijing lineage.
The Beijing lineage.
It's widespread in Asia and has been linked to outbreaks all over the world.
Wow, it sounds almost ominous.
A little bit, yeah. Research suggests this lineage has a higher mutation rate than other strains, making it more adaptable and more likely to develop drug resistance.
So it's constantly evolving, trying to outsmart us.
You could say that. One particular branch of the Beijing lineage, known as Strain W, was responsible for a big outbreak in the U.S. a few decades ago, centered in New York City.
Oh, wow. I remember hearing about that. It's a stark reminder that TB is definitely not a disease of the past.
Exactly.
So we've talked about the bacteria, how it spreads and how it causes disease. But now I want to dive into the world of laboratory diagnosis. That's where you medical microbiologists really shine, right?
That's our domain.
So let's talk about how labs are organized to handle this challenging bug.
Well, a lot of countries use a three-tiered system. You've got local labs that can handle routine testing, like smears and cultures. But for more specialized tests, like species identification or drug susceptibility testing, they'll send those isolates to regional reference labs.
Makes sense. A tiered approach to make sure everything runs smoothly and efficiently. And then there are the national reference laboratories, right?
Yep, the top tier. They handle the most complex testing, do research on new diagnostic methods, and basically oversee the whole network of labs to make sure everything is up to snuff.
A well-oiled machine. And I imagine in recent years there's been a growing need for more specialized mycobacterial facilities, especially with the rise of MDR-TB.
Absolutely, especially in areas where drug resistance is prevalent and in hospitals that care for a lot of immunocompromised patients.
Right, because those patients are particularly vulnerable to TB.
Exactly.
So it all starts with the specimens, right?
Right.
Sputum, biopsies, CSF. Getting those samples to the lab properly is crucial.
Couldn't agree more. Proper collection and transport are essential. We want to minimize contamination and make sure those mycobacteria have the best chance of surviving the trip.
And sputum, I'm guessing, is the most common specimen, but also the trickiest to collect.
You got it. Not everyone can produce a good sputum sample on demand.
I can imagine.
So sometimes we have to get creative. Early morning gastric aspirates, induced sputum, we have to adapt.
Resourcefulness is key. So once those specimens arrive in the lab, what happens next?
Well, we handle them differently depending on where they come from. Samples like sputum, which normally contain bacteria, need to be decontaminated to get rid of those normal flora.
Decontamination. That sounds like a delicate process.
It is. You have to kill the contaminants without harming the mycobacteria we're trying to grow. Once we've got a clean specimen, we can move on to culture.
Culture, the gold standard for TB diagnosis. What kind of media do you use to grow those slow-growing mycobacteria?
We've got a few options: egg-based media, agar-based media, and liquid media. Liquid media, especially when used with those automated systems, can often give us faster results.
Automation is a lifesaver in a busy lab.
It is. Some systems can even monitor growth continuously and alert us as soon as there's a positive culture.
That's incredible. So we've got our culture growing, but we still need to confirm it's actually M. tuberculosis. What are the next steps?
Well, traditionally, we'd use a variety of phenotypic tests to identify the species, but those can be time-consuming. So these days, we're relying more and more on molecular methods.
Molecular methods. DNA detectives. Tell me more.
Well, there are tests like the AccuProbe, which can identify common mycobacterial species within a matter of hours. And then we have commercial systems like INOLIPA that can give us a more comprehensive identification, usually within a couple of days.
Wow, that's fast. What about sequencing? Has that become part of routine TB diagnostics?
It's becoming more common. Sequencing that 16S RNA gene can help us identify any mycobacterial species, even those we've never seen before. And then by targeting other genes like GIRA and RPB, we can really fine-tune our identification.
So you've got this whole toolbox of molecular techniques at your disposal. It's amazing how far we've come. But identification is just the first step. We also need to know which drugs are going to work against that particular strain, especially with drug resistance on the rise.
Absolutely. Susceptibility testing is crucial. And while we still use some of those traditional methods, molecular techniques are playing a bigger and bigger role.
Molecular susceptibility testing. That sounds interesting. So can you reliably predict resistance just based on a bacteria's genes?
For most anti-TB drugs, it's still a bit tricky. Those resistance mechanisms can be really complex, involving multiple genes and pathways.
So it's not a simple one-to-one relationship between a specific mutation and resistance to a drug.
Right. But there is one notable exception: rifampicin.
Ah, our trusty rifampicin. What makes it so special?
Well, remember how we talked about over 90% of rifampicin resistance being caused by mutations in that one small region of the RPB gene?
Yes. It's like a bullseye, that gene.
Exactly. So we can use techniques like direct sequencing or commercial kits that target that specific region to quickly identify rifampicin resistance. It's been a game changer, especially for managing suspected MDR-TB cases.
It's amazing how molecular techniques are really transforming our approach to TB diagnostics. It's like we're able to unlock the secrets of these bacteria and understand them at a whole new level. But I imagine even with all these advancements, there are still challenges, right? We're still human after all, and mistakes can happen.
Of course, even with the best technology and the most skilled microbiologists, errors can happen. That's why quality control is paramount.
Quality control, the foundation of a reliable lab.
Exactly. We've got to maintain that less than 1% cross-contamination rate.
A constant battle against those invisible invaders.
It is. We participate in external quality assessment schemes to make sure we're meeting the highest standards.
So it's like having an outside expert come in and make sure everything is running smoothly.
Exactly. And it helps us identify areas where we can improve.
Continuous improvement, always striving for excellence. That's fantastic. But I'm curious, even with perfect techniques, what are some of the inherent challenges you face in diagnosing TB?
Well, there's the issue of interpreting those nucleic acid amplification tests, those NATs.
All right, those DNA detectives.
They're great for respiratory samples. But when we're dealing with non-respiratory samples, the bacterial load can be much lower.
So the test might not be as sensitive.
That's right. So we have to be very careful about how we interpret those results. And then there's the challenge of molecular susceptibility testing.
Because as you mentioned, for most drugs, it's tough to definitively say this mutation equals resistance.
Right. It's not always that straightforward. But luckily, rifampicin is different. That RPB gene is our beacon of hope.
Or a reliable indicator of resistance.
Exactly.
So we've talked about the bacteria, the epidemiology, and the challenges of lab diagnosis. It's clear that M. tuberculosis is a formidable opponent, but it also seems like we're making progress in understanding and combating it. Where do we go from here?
Well, next, I think it's time to take a trip back in time. We'll explore the history of TB treatment, from the pre-antibiotic era to the development of modern drug regimens, and then we'll delve into strategies for preventing and controlling this persistent threat. There's still so much to uncover.
Sounds fascinating. I can't wait to hear more. Stay tuned, everyone, as we continue our deep dive into the world of Mycobacterium tuberculosis. Welcome back to our deep dive into the world of Mycobacterium tuberculosis. We've covered a lot of ground already, from the bacterium's unique characteristics and sneaky survival tactics to the complexities of lab diagnosis. Now let's step back in time and explore the evolution of TB treatment.
It's quite a journey, that's for sure, from ancient remedies to modern drug cocktails.
I'm ready for a history lesson. Let's start with the pre-antibiotic era. What did people do before those miracle drugs came along?
Well, before antibiotics, options were limited, to say the least. One prominent approach was the sanatorium movement.
Sanatoriums. Those were like hospitals specifically for TB patients, right?
Exactly. Patients would be isolated in these facilities, often located in scenic, mountainous areas where they'd receive plenty of rest, fresh air, and a nutritious diet. The idea was to boost their overall health and give their immune systems a fighting chance.
It sounds almost idyllic.
Oh.
But did it work?
Surprisingly, it did help some patients. The emphasis on rest, good nutrition, and clean air likely contributed to improved health and a stronger immune response, allowing some individuals to naturally fight off the infection.
It's amazing how those basic principles of health and well-being were recognized even back then. But I imagine the discovery of antibiotics completely transformed the treatment landscape.
Absolutely. Streptomycin, discovered in 1944, was a true game changer. It was the first drug that could actually effectively kill Mycobacterium tuberculosis, offering hope for a cure that had previously seemed out of reach.
Streptomycin, a historic moment in the fight against TB. But I'm guessing, as we've discussed before, antibiotic resistance is a persistent challenge. Did resistance to streptomycin emerge as well?
Sadly, yes. Resistance to streptomycin did emerge, particularly when it was used as a single drug. This highlighted a crucial principle that we still follow today: the importance of combination therapy.
Combination therapy. Using multiple drugs simultaneously to target those bacteria from different angles and prevent them from developing resistance. What other antibiotics joined the fight?
Well, PAS, or para-aminosalicylic acid, was another early drug, but the real game changers, the ones that truly revolutionized TB treatment, were isoniazid, discovered in 1952, and then rifampicin, which came along in the 1960s.
Isoniazid and rifampicin. Every medical microbiologist knows those names. How did these drugs change the game?
They significantly improved treatment outcomes and maybe even more importantly, allowed us to shorten the duration of treatment. We went from years of sanatorium confinement to just months of treatment at home.
It's remarkable. From isolation to outpatient care, all thanks to these powerful antibiotics. But I'm guessing ensuring patients completed their full course of treatment became a new challenge.
Absolutely. Imagine you're a patient and you start feeling better after a few weeks of medication. It's tempting to think you're cured and stop taking those pills.
I can see how that would happen. You feel better, so you assume you're good to go.
Exactly. But the problem is, those sneaky TB bacteria can be lurking, waiting for their chance to strike back. If you stop treatment too soon, they can come roaring back. And they might even develop resistance to the drugs, making them much harder to treat the next time around.
That's a scary thought. So how do we combat that? How do we make sure patients stick to their treatment plans?
That's where DOTS, or Directly Observed Therapy short course, comes in. It's a simple yet incredibly effective strategy.
DOTS. We've talked about it before, but I'm always eager to learn more. Can you refresh our memory on why it's so important?
Absolutely. It's all about ensuring patients take every single dose of their medication as prescribed. It involves having a healthcare worker, a pharmacist, or even a trusted family member directly observe the patient taking their medication each time.
It's like having a dedicated cheerleader in your corner, supporting you every step of the way and making sure you finish that course of treatment and conquer TB once and for all.
I like that analogy. And speaking of challenges, we know that drug resistance continues to be a major hurdle in our battle against TB. Managing drug-resistant TB, especially multi-drug-resistant TB or MDR-TB, is no easy feat.
MDR-TB, resistant to at least isoniazid and rifampicin, two of our most powerful weapons. It sounds like a nightmare scenario.
It can be. The stakes are much higher with MDR-TB. The mortality rate is significantly higher, and treatment regimens are longer, more complex, and often involve drugs that are more toxic. It's a whole different ballgame.
So how do you even approach managing these complex cases?
It requires a highly individualized approach.
Yeah.
We need to create a treatment plan specifically for each patient, taking into account the drug susceptibility profile of their particular strain, their overall health status, and the potential side effects of the various drugs. There's no one-size-fits-all approach.
Individualized care. It makes sense, recognizing that each patient is unique.
Yeah.
But what are some of the key principles that guide your decision-making in these challenging MDR-TB cases?
The first and most crucial step is knowing the enemy. We need to know exactly which drugs the bacteria are resistant to, and that's where in vitro susceptibility testing comes in. It's absolutely essential. No guesswork allowed.
Data-driven decisions. Love it. What else is important?
We stick with the principle of combination therapy, but we make sure the regimen includes at least 3 drugs that we know the bacteria are susceptible to. And in areas where drug resistance is widespread, we often start with a 4-drug regimen right from the beginning.
Hit them hard and hit them from all sides.
Yeah.
Makes sense. And I'm assuming DOTS is even more important in these cases.
Absolutely. DOTS is crucial for ensuring adherence to these complex and demanding treatment regimens. We need to support patients every step of the way, helping them navigate the challenges and stay committed to their treatment plan.
DOTS, it's a lifesaver, for sure. Are there any other considerations when managing MDR-TB?
Yes. We have to be incredibly careful when adding new drugs to the regimen. We only add them in pairs, and only if we have solid evidence, usually from susceptibility testing, that the strain is actually susceptible to both drugs. We don't want to inadvertently make things worse by fueling further resistance.
It's like a high-stakes chess match, trying to outmaneuver these incredibly adaptable bacteria. But shifting gears a bit, let's talk about prevention and control. Treating TB is essential, but preventing it in the first place is even better, right?
Absolutely. TB control is a multifaceted challenge that requires us to tackle the problem from all angles. And one of the most important things we can do is address those broader social and economic factors that contribute to the spread of TB.
It's a reminder that TB isn't just a medical issue, it's a societal issue.
Exactly. Poverty, overcrowding, and malnutrition all create breeding grounds for TB. Addressing these issues is crucial.
It's about creating healthier communities where TB has fewer opportunities to take hold. What other public health interventions are important for TB control?
Early identification and treatment are key. We need to find those infected individuals before they have a chance to spread the disease further. That's where contact tracing, active case finding, and screening programs all play vital roles.
It's like a game of hide and seek, but the stakes are much higher. What about access to healthcare services? I imagine that plays a crucial role as well.
Absolutely. Ensuring people have access to high-quality diagnostic and treatment services is paramount. Rapid molecular tests, drug susceptibility testing, directly observed therapy, these are all essential components of an effective TB control program.
Access to care. It's a fundamental human right, and it's essential for controlling TB. What about infection control, especially in healthcare settings where the risk of transmission can be higher?
Infection control is critical. We need to have those robust procedures in place to protect both patients and healthcare workers, particularly in those high-risk environments.
I'm guessing those precautions are even more stringent for patients with suspected MDR-TB.
You bet. We have to assume they're infectious until proven otherwise. Isolation in negative pressure rooms, personal protective equipment for healthcare workers, rapid diagnostics to confirm the diagnosis and rule out drug resistance. These are all standard practice. We can't take any chances.
It sounds like a whole different level of precaution. And what about vaccination? Doesn't the BCG vaccine offer some protection against TB?
BCG has definitely been a valuable tool in the fight against TB, especially for protecting children from those severe forms of the disease. But its effectiveness can vary quite a bit, and in some parts of the world, it's not routinely used.
The ongoing debate about BCG. It's the bottom line.
Well, it's a tool in our arsenal, but not a perfect solution. The search for new and more effective vaccines continues. Hopefully someday we'll have a vaccine that can truly conquer this ancient foe.
It's inspiring to see that ongoing dedication to research and development. The fight against TB is far from over, but with continued research and collaboration, I'm hopeful that we'll eventually see an end to this devastating disease. Well, I think it's safe to say we've covered an incredible amount of information in this deep dive.
We've barely scratched the surface, but hopefully we've given our listeners a good overview of this fascinating and challenging bacterium.
From the bacteria's intricate structure and its remarkable ability to adapt and evolve to the complexities of lab diagnosis, treatment, and control, Mycobacterium tuberculosis is a force to be reckoned with.
It's a bug that demands respect and constant vigilance.
And to all our listeners, thank you for joining us on this journey into the world of Mycobacterium tuberculosis. Keep those minds curious and those microscopes focused. Until next time, stay well.